So that was our introduction, right? Now we're going to go into specifics, right? And this one's kind of hidden in your book. So I'm going to show you how to find it. And I probably should make a note here, because it took me a minute to find it this morning. This animation is a good one, and I don't know if... Where's the book? There's the book. So we're on chapter 20, which is adaptive, right? We're on, so remember mine go by section, so we're on section 2, right? So notice I clicked on section 2, right? So this is section 2, right? We're going to focus on T cells, right? And so there are 2 subsets, right? 2 choices, cytotoxic, helper, or CD8 cytotoxic. So we'll go over that, right? And the special handshake they do. So in here, right here, this cell-mediated animation is a really good one. So let's see, we'll pull it out. I think if you do this, it goes big screen, right? Yes. The arrow means big screen.
Cell-mediated immunity.
This one's somewhat interactive.
Carried out by the
and of course, I still haven't figured out how to get the damn audio to work in this room.
The body contains millions of different T cells.
We have this set of T cells, right? And we know each one of them is going to make their own unique receptor. And this is done before they even see antigen, right? So we produce a million different ones, right?
Which is able to produce millions of variations.
This thing is slow.
Is capable of binding to a
right, so
specific
each one of these is going to be
antigen,
the specific antigen.
But only after the antigen
the problem
has
here
been
is,
and presented by some other cell.
Other than this is slower than me.
Lymphocytes recognize such presenting cells by means of another surface.
We have 2 different types of T cells. So then they're showing the little co-receptor here, right? So this is to represent one type and this is to represent another. So this is CD4, which we call what type of T cell? Helper T cell. Remember, 4H. And then cytotoxic T cells carry the CD8.
And CD8 T cells.
This is in addition to the receptor that actually is going to bind to the antigen.
Most cells that display CD4.
So we'll start out with the helper cells. So here's a poor cell that gets infected by a virus. Antigen presenting cell comes along. So who is that? Start repeating this to yourself numerous times. Dendritic cell, macrophage, right? Or B cell, okay? Or what they're talking about when they're talking about antigen presenting cells. So in this case, it's probably a macrophage or a dendritic cell. So notice it took in and digested. It did that really fast, right? The fragments. And then this is MHC that it's going to load those onto. Notice this was a virus, right? It got inside of our cells. So the presentation here
these protein molecules are only
is on MHC class 2,
called antigen presenting
right?
cells.
But that was something that get inside of our cell, and something you might have not seen in the animation is that, look, it didn't pick it up from the cell. It picked it up from outside the cell. Do y'all see that? That's an important point. Okay? No, it hadn't probably got into that cell yet. Or maybe it just got released by a neighboring cell, right? But it grabbed onto it, right? It gobbled it up, broke it into pieces, loaded it onto MHC. Now it's going to do what we call antigen presentation. It's going to present it to that T cell. So the T cell comes along, right? Oh, not a match. This one, on the other hand, is a match. Notice that the antigen is binding to the T cell receptor. But it's the MHC that's actually presenting it. What is binding to the CD4? It's the MHC. So this is where I say it's like a secret handshake, where the antigen presenting cell, in this case, right, maybe a macrophage or a dendritic cell, can only talk to this helper T cell, because this is CD4 and this is MHC class 2. See if they draw it in. I can't remember.
Needed to begin activation
T-cell receptor.
A second signal, known as co-stimulation, must also occur.
And they're actually going to talk to each other, right? Cytokines are going to be released. Not only is there secret handshake, there's communication that's happening here.
Provide co-stimulation. Cytokines, small protein hormones.
So look, cytokines released, activates this T cell.
Once the T cell is activated,
so now
it proliferates into clones of itself.
Activation for T cells or B cells or any of these lymphocyte cells. Do you want just one T cell running around recognizing that virus? No, you want multiple ones. So the very first thing is clonal expansion, right, or proliferation. Clonal, why? Because all these guys are, what's clone mean? They're the same. All these guys are the same. Do you see that? All have the same receptor on them. They're all going to recognize the same virus, right? So those guys are going to go out and hunt down these virus-infected cells.
The receptors on these cells are able to bind.
Now they become what we call effector cells, the ones that go out and hunt. I should have paused right there. But some of them become memory cells that hang out in your lymph nodes and your lymphoid tissue in case, guess what? The bad guy comes back again. Those who get activated when it's your second exposure or any exposure past primary, right? Why we call it memory. So effector cells, as their name implies,
helper T cells secrete
are going to do something. Helper T cells,
is interleukin 2.
Guess how they help?
In most immune
I call them talker T cells.
It is also known as T
That's my nickname for them, because they're going to release cytokines like IL-2, which is going to tell other cells what's going on, right?
Other cytokines
or IL-4, right? What are all of these? IL stands for interleukin. It's a white blood cell talking to a white blood cell, right? Or
specific resistance.
Interferon, right? When we've got viruses talking to, yet again, say a macrophage.
In general,
the good news for you guys, the only cytokine we're going to concentrate on is this first one.
Subsets of helper
a little bit later on. Okay. So one of the cells they do help is that cytotoxic T cell. That's the cell that has the receptor on MHC class 2.
Healthy cells don't survive
class 1.
In the thymus gland. Surviving
God,
T
different
cells
classes are driving me nuts today.
That
if this cell is presenting and that cytotoxic T cell recognizes, this is when it kills, and this is how it kills. It releases toxic substances onto that cell that will lyse that cell. If it's a match, right?
Secreted by cytotoxic T cells.
So that's a good animation to walk through slowly, but don't get caught up in all the talking that the helper T cell does. So we'll close that one. That one's definitely a highly recommended one. Unfortunately, kind of hard to find in the book. So that one's titled cell-mediated. There are some components of what we would call humoral. So what do we need to know? What are these things doing, right? What's that interaction? How are T cells activated? Those co-receptors, the CD4 and the CD8, which determines whether it's a helper or a cytotoxic T cell. How are those MHCs involved, right? So we can step through that. T cells are involved mainly when we talk about cell-mediated. You've got to have T cell involvement. You've got to have antigen presentation, as we saw, right? You've got to have a cell pick up the antigen and present it to that T cell. You've got to have that cell signaling. You've got to get that co-stimulatory molecules that they didn't show interacting. This is a pretty involved process. But once activated, those T cells, as you saw, are going to proliferate, right? So we have a whole army now that specifically recognize that antigen. And some of them are going to go out and fight. Those are the effector cells. Some of them are going to become memory cells. Right? So, naive. What do we mean by naive? Hasn't been exposed to an antigen yet, right? This has been born maybe from the bone marrow, it's been educated in the thymus. It isn't until it encounters antigen, which is not a step shown here, hence the highlight, right, that they've been activated, that we saw in that animation, that they're going to clonally expand and differentiate. Some of them become effector cells, right? They go out and do their job, and once their job is over, look, they're short-lived cells. They're going to go through a process of cell death known as apoptosis, right? When we're done with them, we're like, okay, we don't want to feed you anymore, house you, you're expensive. We're done with you. Because we're going to keep these guys around. Yeah. All right, so who's going to present to those T cells? Antigen presenting cells. But really the specialists. Who are our 3 specialists that we need to remember? Hence why it's blocked out, so you'll write it 5 or 6 times. Macrophages is 1. Dendritic cells, which we haven't really talked much about yet, but we will. And B cells, which also are known as B lymphocytes, which again we're going to save to the end. So here is an antigen presenting cell. We're not being specific as to whether it's a specific antigen presenting cell or not, right? We're not saying what class MHC. Do you see that? But we're loading a foreign protein into it and we're presenting it on the surface so that a T cell receptor can bind to it. This is a very general drawing, right? So again, T cells have their own specific one. They mature in the thymus. In the thymus, they're going to be screened. We don't want anybody who's recognizing self, right? We want to get rid of those. We want to make sure their receptors are good, right? Then we're going to send them on their way through the blood to go to their homes, what we call as secondary lymphoid organs, right? Things like spleen and lymphoid tissue. So starts out in the bone marrow, travels to the thymus, and then we get all these beautiful T cells, right? And notice this antigen presenting cell is actually even interacting with 2 different T cells. 2 different subsets of T cells are going to be activated here, right? Each recognizing a different antigen. All right, so if I say this is MHC class 1, that means this is a cytotoxic T cell. And notice signaling happens all the way to the nucleus. We get gene expression, we get cytokines released, right? We're going to get communication to the system as to what's going to happen. But if this is MHC class 1, this has got to be a cytotoxic T cell. Because remember, class 1's found on all our cells, right? So if any of our cells gets infected, we want to know. And what do you want to happen to that cell if it gets infected? You want it to die, right? So remember, cytotoxic T cells have CD what? 8, because it sounds like eat, right? So MHC class 1, right? The very first one found on all nucleated cells. This is our first line of defense, right? If something bad gets in, virus, bacterial. If we can present, we have an activated T cell, right? Then it's going to kill that cell. Now when we move into the specialists have MHC class 1, but they also have 2. So these are your macrophages, your dendritic cells, and your B cells. These are cells that actually can take stuff in from the outside, something we're going to talk about next time. And you saw in that animation, right, where he actually gobbled up the virus. Well, because he gobbled up the virus from outside of a cell, he presented it on MHC class 2 to a helper T cell. Which, remember, these are the real talkers, right? They're going to release a whole bunch of cytokines. They even talk to themselves. Right? So helper T cells, it's CD4. Remember, 4H. So CD4, remember, synonymous with helper T cells, MHC class 2. Where CD8, right, is our first line of defense, MHC class 1, all nucleated cells. They're going to kill them if they've been activated and they're presenting. And again, that signaling is going to only elicit certain genes, right, certain information in the nucleus to be activated so that the signaling, hopefully, is the correct choice. Right? And the one magic cytokine is IL-2. This is the big cell division one, right? This is the one that really, when we get activation, is released and causes those cells to proliferate and go through the cell division cycle. So this is that snapshot of that video, right? So again, just to remember that helper T cells are going to do a lot of cytokine release. And different cytokines are going to talk to different cells in different ways. Effector cells go out and do their job, right? They're going to do something. So in the case of cytokines, right, secretion of large amounts of cytokines. Helper T cells, they're big time talkers. Where cytotoxic T cells, as their name implies, right, they're going to go and attack natural killer cells, but in this case, it's a receptor-to-receptor interaction. Natural killer cells kill if you don't have MHC class 1, or you're covered in an antibody that it recognizes. But they both kill in the same way, right? They're going to release granzymes, perforin. They're going to initiate apoptosis, program cell death of that cell. Memory cells, right? Much longer lived. When they're activated, it's faster, it's more aggressive, right? And this happens anytime after primary, right? Once you get primary response and you get memory cells, anytime after that, we're going to initiate that response. I know. This is my last slide. This is just a diagram, right? Is showing you those interactions. So we'll maybe come back to this one next time because we're out of time.